All right, we're a couple minutes past the hour. Good to see everyone. Happy New Year. This is our first event of the year. And today I would like to welcome Ariadna Tibau Martorell, who is a medical oncologist and health services researcher from Barcelona. And currently she's working with Aaron Keselheim's team in Boston. And it's a portal team. We've had Aaron on a few of our events. And Ariadna published a very interesting paper. We've been talking about cancer on many, many events. And of course, the issue of access to medicines and cancer care, especially in the US, is a very difficult one in terms of imbalance of the benefits of new drugs that new drugs provide and the exorbitant costs. So I'm inviting Ariadna to share their research and her past work on the subject. So Ariadna, please, the floor is yours.
So thank you, Lisa and Jake, for this kind invitation. Welcome, everyone, and thank you to all of you for being here today. I'm excited to talk about something currently central to cancer care, which is timely access in cancer care and defining clinical benefit and value. Cancer treatments are advancing quickly, but the real challenge is making sure patients get therapies that truly improves their lives, not just by extending survival, but also by enhancing quality of life. In this talk, I will focus on how we define clinical benefit and value in cancer care and how we can balance this need for quick access with meaningful outcomes. So let's get started. Here are my conflicts of interest. My presentation is divided into 4 sections. The first covers cost. The second address clinical benefit. The third explored the intersection, so value in oncology. And finally, I will talk about timely access to cancer and why it matters. This slide shows global pharmaceutical spending by region using, in recent years, the cost of cancer drugs has risen sharply. In Europe, oncology cancer drugs cost grew from 10 billion euros in 2005 to over 3 times in that amount by 2018. Similarly, in the United States, cancer care spending increases by an estimated 5-7% annually. When we analyze when we see the global pharmaceutical spending by region using IQVIA data from 2019 to 2023 and projections for 2024 to 2028 in US billion dollars, what we see is that emerging markets are growing rapidly. However, developed markets, USA, UK, Europe, remain the largest contributors to this cost. In recent years, a better understanding of cancer biology has led to the discovery of new therapies, some of them showing unprecedented responses in early trials and improved survival outcomes in later studies. In this context, regulatory changes allowing rapid review and approval of cancer drugs with unmet needs, along with streamlined development approaches, have increased the use of surrogate endpoints for approval, supporting approval. These 3 factors combined have been key in the search of the FDA cancer drug approvals. The rate of approvals has risen sharply in the 21st century, with the average annual approvals increasing from 7.4 per year between 2000 and 2004 to 56 per year from 2017 to 2022, an impressive 757% increase. As shown in the next slide, we are moving from a higher level of certainty about benefit with regular approval to a lower level of benefit with accelerated approval due to the endpoints that support the approval. Overall survival is the gold standard and the preferred endpoint for regular approval, as it directly measures benefit. In contrast, response rates, an endpoint that doesn't reflect how patients feel or how long they will survive, is supporting and it's commonly used for supporting accelerated approvals. This changing approach has allowed accelerated patient access to new treatments. However, the trade-offs for rapid approval include, first, the increased use of intermediate and unvalidated surrogate endpoints, even when they haven't been proven to reliably predict overall survival or quality of life. Second, the under-recognition of adverse events at the time of market entry. And finally, a more limited understanding of clinical benefit. Due to rising clinical prices and the growing number of new drugs, many relying on improvements in surrogate endpoints, the definition of clinical benefit has become increasingly important in recent years. Why is clinical benefit important? As oncologists, we spend considerable time discussing treatment options and potential adverse events with our patients. Although the complexity of treatments has increased over time, what really matters to patients remains simple. They want to live longer and to live better. Thus, the FDA and EMA criteria for drug approval requires substantial clinical evidence of clinical benefit or efficacy from adequate and well-controlled trials, and efficacy should be demonstrated by improving overall survival, improving quality of life, or a proven surrogate of these endpoints. The following question is, how do we define the clinical benefit of a treatment? Clinical benefit is the balance between an efficacy endpoint, or preferably overall survival, quality of life, and the drug's toxicity. And value in oncology refers to the balance between clinical benefit and financial cost, aiming to find the best treatment for a given clinical scenario at the lowest cost. To increase value in cancer treatment, several organizations have developed scales to standardize clinical benefit. The most commonly used in the literature are the American Society of Clinical Oncology Value Framework, or ASCO Value Framework, the European Society for Medical Oncology Magnitude of Clinical Benefit Scale, ESMO-MCBS, and the National Comprehensive Cancer Network, NCCN, evidence blocks. These frameworks, all of them, consider a combination of benefits measures, including efficacy, safety, and quality of life. Probably among them, ESMO-MCBS stands out for several reasons. Has had regular updates, the latest one in 2022. It provides expert scores for all FDA and EMA-approved cancer drugs, most of them available online with the studies and explanations. and performed by experts. It applies to both solid and hematological liquid tumors and evaluates both randomized clinical trials and single-arm studies. The ASCO value framework has the handicap that excludes single-arm trials for their analysis, relying only on randomized clinical trials, which limits its relevance nowadays that the number of drugs approved based on single-arm studies is increasing. And the NCCN evidence blocks, although includes information about the cost, was just introduced in 2016 and ranked treatments based on expert opinion, which can be an important bias, and considers the available evidence at the time of guideline publication, but it's not focused on individual trials. As I mentioned, the ESMO MCVS is a composite measure of clinical benefit that evaluates drug efficacy, safety, and impact on quality of life. This tool aims to quantify the clinical benefit of new therapies and is used to help patients, physicians, and health technology assessment choose high-value treatments. So it helps prioritize cancer medicine. The framework uses different forms for curative and non-curative setting. And as you can see in this slide, in the curative setting, ESMO MCBS grades range from A, high, to C, low. And while in the non-curative setting, they range from 1 to 5, being 4 and 5, the substantial clinical benefit scores. Several groups have analyzed the value of cancer drugs approved by the FDA and EMA using these tools in a series of studies conducted in collaboration with Dr. Eitan Amir from Princess Margaret Hospital in Toronto. We aim to answer the following scientific questions. Which is the clinical benefit of approved cancer drugs? Which is the clinical benefit of cancer drugs assessed in single-arm trials as the basis for approval? And what is the long-term clinical benefit of approved drugs? We applied the original version of the ESMO MCVS scale, which only allows for the evaluation of randomized clinical trials, and the current version 1.1, which permits the evaluation of randomized trials and single-arm trials, to 105 randomized controlled trials and 27 single-arm trials that supported FDA approval of 63 solid cancer drugs in 118 indications between 2006 and 2016. Using the original version of the scale, the results show that only 44% of the treatments reach the ESMO MCVS threshold for substantial clinical benefit. Notably, over time, there was a statistically significant improvement in the number of trials meeting the ESMO threshold for substantial clinical benefit, and partially due to improvements in quality of life and or the presence of fewer severe toxicities. As you know, the observation of high and long-lasting responses to targeted therapies has raised questions about the need for traditional approval based on randomized clinical trials. When applying the ESMO MCVS version 1.1 scale to the same cohort, about 1/3 of trials supporting FDA drug approval met the threshold, about 1/3, met the threshold of substantial clinical benefit. And a small proportion of single-arm studies, 7%, only two of 2 out of 27 studies met this threshold. This included osimertinib for T790 mutations mutated in non-small cell lung cancer, and crizotinib for ALK rearranged non-small cell lung cancer, both scoring a 4 on the ESMO scale. These results are in some ways expected, as single-arm trials must demonstrate not only a significant response rate and a long duration of response with the scale, but also need to include data from quality of life and post-marketing studies to meet the substantial benefit threshold. The low proportion of single-arm trials meeting this threshold may reflect a lack of supporting data, or some authors may say that higher standards set by the ESMO MCBS. One following questions is, must some trials show benefit post-approval? As a way to answer this question, what we did is that we evaluated the same cohort of cancer drugs approved by the FDA, but covering only the period between January 2006 to December 2015, with post-marketing follow-up until April 2019. Trial-level data supporting approval and updated overall survival and quality of life was reviewed. And in this occasion, we applied not just the ESMO MCBS scale, but also the ASCO Value Framework version 2 to both initial studies and follow-up studies. Our findings showed that compared to the time of approval, most anti-cancer drugs improved overall survival, more anti-cancer drugs showed an improvement on overall survival and quality of life, and meeting the ESMO MCBS or ASCO value frameworks thresholds for substantial clinical benefit during this post-marketed period. However, only about a half of trials achieve the substantial benefit threshold. And companion diagnostic tests and immunotherapies were those associated with greater clinical benefit over time. An area of particular interest today is precision medicine. The number of genome-targeted cancer drugs has risen, providing the opportunity for personalized therapy, but often at very high cost. At present, genome-targeted therapies covers around 40% of new and supplemental cancer drug indications, and these therapies are defined as drugs approved based on genomic tests in which the drug targets a given genomic alteration. This approach allows for personalized treatment, enhancing efficacy for patients who test positive and avoiding toxicities for those who test negative. Examples of groundbreaking treatments are trastuzumab pertuzumab for HER2 breast cancer and osimertinib for EGFR mutated non-small cell lung cancer. However, their benefits are limited. The benefits of genome-targeted drugs are limited for most patients with metastatic cancer, mainly due to the small number of patients who respond to the treatment and the challenge of drug resistance and cancer progression. So as shown in the figure, on the left side on the slide, less than 5% of cancer patients in US were estimated to benefit from genome-targeted drugs. Advances in next-generation sequencing allow for the rapid sequencing of large number of nucleotides. However, to date, most genomic alterations are of unknown clinical relevance. In this context, and given the uncertainty about the validity of the targets and the clinical benefit of some of these drugs, most of them approved based on surrogate measures, we asked two key scientific questions. How valid are the targets and the clinical benefit of FDA-approved genome-targeted cancer drugs? And how valid are these targets and the clinical benefit when are recommended in clinical practice? Before going into the studies, I would like to a little bit explain how address these questions. To address the challenge of value in precision medicine, a working group from the European Society for Medical Oncology created a classification system that ranks genomic alteration and targeted cancer therapies based on their level of evidence and clinical relevance. This system is called ESMO Scale for Clinical Actionability of Molecular Targets, or ESCAT. And as shown on the slide, genomic alterations are classified into 10 tiers from class 1A to class 10 with decreasing clinical benefit or decreasing clinical relevance. The goal or the aims of the scale is to identify actionable genetic alteration in cancer therapies and help guide decisions about using molecular targeted therapies. The SCAT system considers two key factors. First, the level of evidence, which reflects the study design of the clinical trial in which the biomarker is analyzed. And second, the clinical relevance, which refers to the specific treatment setting and indication. I won't go into detail about every level of evidence, but I will give you a brief overview. Tier 1 alterations are clinically relevant and ready for be used in practice because clinical trials have shown a significant survival benefit with the combination of the genetic alteration and the targeted therapy. Tier 2 are alterations with a potential clinical relevance being used on retrospective or prospective trials that did not show a survival benefit, overall survival or a PFS benefit or disease-free survival benefit. Tier 3 targets are linked to expected improvement in outcomes based on trials in other tumor types. Tier 4 show promise in clinical and preclinical studies, and tier 10 alterations lack evidence as therapeutic targets and should not be used in clinical decisions medicines. For example, germline BRCA1 and BRCA2 mutations as targets for rucaparib are classified as tier 1A in ovarian cancer, tier 1B in prostate cancer, and tier 2B in pancreatic cancer. And this classification is based on the knowledge from clinical trials in different tumor types. Another important point about the scale is that originally, the SCAT framework was a system focused on clinical benefit, ranking genomic alterations ready for routine use, SCAT evidence class 1A and 1B, as targets for cancer precision medicine, linked to meaningful improvements defined by the ESMO MCBS scale. More recently, the magnitude of benefit is not specifically considered, so ESMO guidelines give a level evidence 1A or 1B to drugs that don't match with a substantial clinical benefit in the trials supporting the approval. So based on this background and these scales, what we did it is that we evaluated the validity of molecular targets and therapeutic benefits of FDA-approved genome-targeted cancer drugs based on the results of the pivotal trials supporting the indication. We analyze all genome-targeted cancer drugs approved by the FDA between 2015 and 2022, and we apply to the SCAT and ESMO MCVS scales to these drugs. We defined high-benefit genome-based cancer therapies by identifying molecular targets associated with substantial clinical benefit as considered by the ESMO MCVS and qualifying for SCAT category levels 1A and 1B. Here a summary of our results. We identified 50 molecular target drugs covering 84 different indications. And among these, 29% were associated with high-benefit genome-based cancer treatments. Using the same scales, we analyzed how these scales apply the clinical practice. In this cross-sectional study, we analyzed genome-targeted cancer drugs recommended by NCCN guidelines in the advanced setting. We collected information about 411 recommendations from these guidelines related to 74 genome-targeted drugs, targeting 50 driver alterations. And as you can see in the slide, in the left side, most recommendations were associated with clinical trials of various types, so were 1A, 1B, 1C, 2A, and 2B, and 3A. However, 16 relied on only case reports or preclinical studies. The SCAT Tier 1 targetability encompassed 60% of target recommendations. And when we combine both scales, as you can see in the right side of the slide, only 12% of trials supported a determination of high benefit genomic-based cancer treatments. So let's talk about the key findings and the policy implications for the first part of the talk. Over time, more trials have started meeting the ESMO MCVS thresholds for clinical benefit, which is a progress. But here is the thing: less than a half of the randomized clinical trials supporting FDA approvals actually meet these standards of clinical benefit. What about approvals based on single-arm studies? They are even less likely to show strong benefits on the ESMO scale, likely because of the scoring thresholds. And another important point is that new evidence demonstrating clinical benefits is rare after regulatory approval. So what does this mean? ESMO MCVS and the SCAT scales can guide and can be valuable tools for guiding cancer treatment decisions. But we think that I think that will be crucial that prescribers, physicians communicate this variability in the evidence supporting drug approvals to their patients. Let's move on to the section on value. Some of the important questions that arise here is, was there an association between clinical benefit and price for cancer drugs approved? And there is a relationship between clinical benefit, launch prices, and post-launch prices changes for cancer drugs. Our objective, our aim on this trial was to evaluate the relationship between the clinical benefit of approved cancer drugs based on established frameworks and their prices in the USA and four countries, 3 European countries: England, Switzerland, Germany, and France. And for this cost-benefit analysis, we identified all new cancer drugs approved for by the FDA between 2009 and 2017, and by the EMA agency up to September 2019. For drugs approved we apply both scales, and we then compare the monthly drug treatment costs with different levels benefits. And what we found is that in the USA, the monthly drug treatment costs were found to be 2.31 times higher than those in the European countries evaluated. And the most important finding was that there was no significant association between monthly treatment costs for solid tumors and clinical benefit across all countries using the ESMO MCBS scale. And similarly, no significant association were found for all countries except France when using the ASCO value framework scale. In a following project, also done in Portal, what we saw is that during the study period for economic evaluation, prices at the time of approval were significantly higher in USA than in the European countries analyzed. At this gap increased in the years following approval. And this increase often was, and the cancer drug prices in USA often rose faster than inflation, while decreased in inflation-adjusted terms in Europe. The second important finding, or that I find that it's relevant, is that price increases at the time of approval and during the post-marketing period were not associated with clinical benefit in any country. So let's break down the key findings and what they mean for policy. We didn't find any strong link between the cost of monthly prices for solid tumors and the clinical benefit. I think it makes sense to prioritize cancer drugs with the highest clinical benefits when negotiating prices. Negotiating the price of the drugs is key for controlling the cost, and frameworks can help with that. In the US, cancer drugs frequently increased faster than inflation, while in Europe they dropped when adjusted for inflation. However, while the US has the biggest challenge in controlling high cancer drug prices, Europe should also think, Europe and UK should also think about reviewing its pricing rules to make sure they match up better with the clinical value of the drugs. And finally, I will talk about timely access in cancer care. At Portal, we have been actively working on various projects related to rapid access in cancer care, especially working on the accelerated approval pathway. However, these projects are still pre-publication and therefore under embargo. I will discuss one of the most beautiful or compelling papers that has been recently published on this topic. Um, this work, led by Dr. Canavos from London Economic School, was recently published at Lancet Oncology. And in this project, the authors examine whether the ESMO MCVS score can be used to predict HTA outcomes and the time to positive outcomes. Um, they they control for a range of variables that had not been considered in previous studies, including clinical and economic uncertainties and considerations beyond cost and clinical effects. For example, they included in the analysis the unmet medical need, the administration advantage, and disease severity. This was a retrospective analysis, and data were extracted from publicly available HTA agencies from six countries, six developed countries: Australia, Canada, England, France, and Quebec and Scotland. And they analyzed non-solid cancer drugs approved in the non-curative setting. As we can see in the slide, this investigation found that high ESMO MCVS scores, 4 or 5, are a significant predictor of positive HTA outcomes. And what also they found is that those drugs achieving high ESMO MCVS scores were approved faster than those showing low benefit, according of the scale. And this finding was maintained in the multivariable analysis when adjusting for other significant predictors of HTA outcomes. Several other factors were analyzed, and some of them also show a significant association to positive HTA outcomes, notably orphan drugs and drugs with high unmet needs, having an active comparator on the clinical trial, having a double-blind study design. And interestingly, or what I find that it's interestingly, is that when there were uncertainties over the magnitude of clinical benefit, this was related to a negative association with HTA outcomes. As a conclusion, I will highlight where I think that future directions, next areas of working, and final conclusions. The ultimate goal or aim of drug development is to provide solid evidence that the treatment delivers meaningful benefits to patients. Value frameworks can play a key role in identifying these therapies with high clinical benefit that should be quickly accessible worldwide. There are natural extensions of these studies, which I think should include the analysis of hematological cancers. Most of these trials didn't include these tumors on their analysis. The assess of treatments designed for curative treatment, that are often also excluded of this analysis. Very important, I think, that explore HTA decision-making for cancer medicines in upper and middle income countries. And I think that prioritize patient perspective and symptomatic improvements through drug development and approval process should be priorities. So I will finish my talk with a huge thanks, a big thanks to Dr. Agustí Barnadas, Consomolto, Aida Bujosa, Thomas Hank, Kerstin Bockinger, and Eitan Amir, and of course, Aaron Kesselheim for the inputs they work on all these projects, and without them, all this work will not be done. So thank you, and thank you all of you for attending this meeting, and I will be very happy to discuss further if there are any questions or that you would like to discuss.
Thank you so much, Ariadna. If you could close your slides, we can get to the Q&A part of the event. So yes, I look forward to your hands for questions to Ariadna. But Ariadna, I have one question for you on the slides where you were presenting launch prices in the US versus Europe. How did you find the prices, or which prices did you take? Because usually they're secret, and the published prices are subject to significant discounts, which are negotiated in confidence. So just a question, where did you take the data from?
No, in this work, I worked more on the part of the clinical benefit and applying the ESMO scale. So I didn't collect this information directly, but was the price reported by regulatory agencies of each of these countries discussing the price of the drug. So it's not the net price. It's not the price considering the rebates that the drugs have. So this is a huge limitation of that we have currently working on these projects.
Okay, no, it's understandable because obviously if someone had this access to this information, it would be a slightly different pricing world. But am I right to assume that the US prices that you compared to were also without particular rebates that different healthcare providers might offer?
So, sorry, may you repeat the question?
Because my question was, I understood what you took for European countries where you took the prices. Am I right to assume that US prices that you used for comparison might also not reflect the actual amounts paid, and they were particular rebates that may happen through different healthcare providers?
Yeah, the price, yeah, it's a very good question and also tricky. The USA prices were collected from the Orange Book, sorry, the Orange Book from USA, and didn't consider any rebates.
Okay. All right. Any questions for Ariadna? Okay, Rita, please go ahead.
Hello, good afternoon. Congrats for the session. I did have one question regarding the SCOT. The change from classifying, from the classification being on clinical benefit to actionability and the evidence supporting actionability. Do you know why it changed? And do you agree with it? Thank you.
Yeah, this is a very good question. Thank you for asking. I think that probably originally when the SCAD was created, probably the expectations of the clinical benefit will be higher, and the reality of many of these cancer drugs are approved. Many of the genome targeted drugs are approved very fast, based on single arm trials or based on randomized clinical trials where the main endpoint is PFS. There are other groups that have demonstrated that just drugs approved based on companion diagnostic tests usually are not approved based on overall survival. So this characteristics linked to the genome-targeted drugs makes that many of them, at the moment of approval, they don't reach the threshold for substantial clinical benefit because they don't show an overall survival benefit, and results on quality of life probably arise later on, or because they are approved based on single-arm trials, impressive response rates, but impressive percentage of response rates, durable, but without any support of randomized clinical trials. So what that means is that most of them, as we show in these two projects, are approved showing as low percentage of high clinical benefits. So most of them will not be scorable if we apply the initial SCAD scale. I'm not sure if I'm answering your question.
You did. My doubt is if we did have the SCAT having the classification based on clinical benefit, maybe in terms of framework and pricing negotiation, maybe the industry would be motivated to move towards targets that have shown more promising results in the past. So we wouldn't have Me Too drugs on actionable genes, but that in the end show no clinical benefit on overall survival. That's why I think, I don't know if changing the SCAT to a lower threshold of quality, if it's good policy, especially in countries in Europe, that in the end it's the citizens that end up paying through taxes the funding. And well, that's just a particular comment, not really a scientific one, but in the end, we should have a framework that prioritizes added clinical value, regardless of the actionability of the genes being demonstrated by thorough clinical evidence. But that's just me. Thank you so much.
Yeah, I'm totally agree with you that lowering the threshold is not a solution and probably makes the solution much more difficult. Totally agree. In fact, in our project, what we show is the result. I didn't go into deep on these two papers because I preferred to give an overview of everything. But in this work, we really split the results, just showing the SCAD without considering the clinical benefit and the results, linking this SCAD scale with the clinical benefit. My concern is that maybe for many physicians and even patients, or the importance of randomized clinical trials in this scenario is less considerate. I mean, not in my case, but I think in the daily day. I mean, and this is what we try to show in the second paper at the BMJ, to apply the same scales in recommendations of the NCCN guidelines that are guidelines followed overall, are the most downloaded guidelines globally. And what we found is that in clinical practice, the recommendations are even less strict in terms of clinical evidence.
I agree. It's a very desperate setting, and you start to lower your threshold for what you consider acceptable, even though you know that ultimately it does not really direct to added clinical value. Thank you so much.
Thank you. Thank you very much.
Joel, great to see you. You wrote a very similar paper for Canada, so please go ahead with your question or comment.
So my understanding is, from what you said, that many of the oncology drugs that are approved on the basis of single-arm trials, which means that when they hit the market, if they are approved, um, the people who are prescribing them don't really know how they compare to existing products. But my question is, even if there are randomized controlled trials, what percentage of those, do you know what percentage of those trials are superiority trials so that when the new drugs come on the market, oncologists have an idea of how they compare to what's out there already?
Yeah, very good question. Okay. Mm, mm, so when we analyze it, FDA drug approvals, what we found is that around I don't know the exact number. I don't remember the exact number, but around 30-40% of the approvals were based on randomized clinical trials. And the design of the study was trying to show a superiority. So around 30-40%. It's true that for this paper, if during the period analyzed, a drug was approved through accelerated approval, but the confirmatory trial, which can be a randomized clinical trial, was a randomized clinical trial, we only considered the randomized clinical trial. So we have been very conservative with this project because we didn't want to duplicate results. I'm not sure if I did answer your question.
Well, just in follow-up then, if the confirmatory trials were randomized clinical trials, how long between initial approval, and the results of these confirmatory trials, what was that period of time? Because if they were initially approved on single-arm trials, then until the confirmatory trials were completed and published, the doctors wouldn't have known how these new drugs compared to existing ones. So do you have any idea of what that time period was?
Yeah. Yes. Okay. The median time that has been reported from the accelerated approval to get so the median time between the accelerated approval and confirmation of benefit and the conversion to regular approval are 3 years, the median time for any drug. And genome-targeted drugs have been shown that this period is shorter. I have the data. I can send you the data. I have not the data in my mind now, but I have the data collected, and I think that probably will be around 2.5 years, 2 years. It's shorter. For the genome-targeted drugs, it's shorter than the median time of 3 years.
All right, thank you. So Ariadna, since I don't see any more hands, a difficult wrap-up question. Where do you see things going from here? Because for as long as I remember being in the field, we only keep talking about this really negative trend, faster approvals, higher prices, more approvals of drugs with non-demonstrated clinical, well, poorly demonstrated clinical benefit or not convincing data. So the trend is completely upward in that direction, and more papers ring the alarms. So does your work. Where do you see that going?
Wow, this is I don't know the answer. Sorry about that. I don't know the answer. I think that probably we will need to really prioritize. So I think that the paper right now say that we need to prioritize, but this recent work presented for in public, the last paper that I presented, published by the London Medical School, what shows is that in fact, regulatory agencies from high income countries that they negotiate the price of the drugs are doing this, prioritizing. So giving more support for rapid approval for drugs achieving high clinical benefit. So I think that this probably this will be the pathway, because we will have the resources to approve drugs showing less benefit. But maybe I'm too much optimistic. I'm an optimistic person, so but I want to think that we will be able to
Turn it
handle
around.
our resources, I hope.
Thank you, Ariadna. So I'd like to give a word to Gonzalo.
Hi, yeah. Can you hear me okay?
Yes.
Yeah.
Yeah, okay, perfect, thanks. So first of all, I really enjoyed your session. I thought it was very insightful and very interesting to have this kind of point of view and such a rigorous work to have been done. I think it's really laudatory. I'd be very interested to know, I mean, many cancer drugs are approved essentially as Me Too compounds. So they don't really have an expectation of having a clinically significant added value based on other compounds that are already on the market. I mean, pembrolizumab and nivolumab are easy examples that come to mind across the many different settings and indications. Did your work account for these Me Too drugs where added clinical benefit would not really be expected?
So, okay, I think that the first thing is thank you for your commentary, and I think that probably when I talked about future directions, I should also include um, more deep research on this topic, because I think that Me Too drugs is an actual I don't know if the word is problem, but I see that the first thing is doesn't helps to control the price, which I think it's important. So competition between Me Too drugs is not helping to decrease the price of the drugs, and also is a way to avoid that the resources are be placed in innovation, in fact. So I think that this is a very important topic, and also think that one of the problems that currently arise and probably also doesn't help to handle with research of high quality is that many Me Too drugs are approved very fast after the first drug approved. So that is also a problem, because most of these Me Too drugs are not compared with the best control arm. So I'm not sure if I'm answering your question, but these are my thoughts about this field.
Gonzalo, do you have any further comments?
No, I mean, I think that's a very, very interesting point of view, and I would fully agree. So I'd like to, echo the words. I completely agree. Thank you.
Thank you, Gonzalo.
Ariadna, thank you so much for this very insightful talk. So many details, even though we keep coming back to the same subject, but discovering new and new points and details and further evidence. So good luck with your further work. So thanks so much for sharing your knowledge with us, and we'll see everyone in 2 weeks. Bye-bye.